What if some of the emptiest stretches of human prehistory are empty only because archaeologists were looking for the wrong kind of evidence? For more than a century, the strongest proof of human presence was a body part, a tooth, a skull fragment, a bone, preserved long enough to be found. Where those remains did not survive, the ground often fell silent. Entire landscapes could seem unoccupied simply because nothing human was left to dig up. That silence is starting to break. In cave floors and buried sediments, researchers are finding ancient DNA shed by humans and animals long after flesh and bone have disappeared. The implications reach far beyond a new laboratory method. If traces of vanished people can survive where skeletons do not, then the old map of human origins may have been shaped not only by where our ancestors lived, but by where their bodies happened to last.
Bones became central to archaeology because they seemed to answer the hardest question cleanly: who was here? A tool could be traded, copied, or carried in from somewhere else. A hearth could be reused, but a tooth was harder to argue with. A fragment of jaw, a bit of skull, the shaft of a long bone, these could be measured, compared, set against other finds, and made to speak in a language the field trusted. From them came species names, body shapes, ages, injuries, and diets. They turned deep time into something almost anatomical, and they lasted, at least sometimes. Teeth especially. They survived where softer tissue vanished, where memory vanished, where everything that made a life recognizable had been stripped away except enamel and shape. So, archaeology learned to build around what endured. The great sites of human origins were not only places where ancient humans had lived, they were places where the dead had remained legible. That legibility hid a bias so basic it could pass for common sense. Human presence is usually light on the ground. A short occupation, a shelter used for a season, a path crossed many times but never settled for long. None of that promises a body. Even where bodies were left behind, the record was fragile. Acidic soils dissolved bone. Water shifted layers. Carnivores dragged remains into darkness or out of it. Exposure, trampling, erosion, later occupations, and the chemistry of a cave floor all played a part in deciding whether a human being would still be there to find. So, the map that emerged was never just a map of where ancient humans had been. It was also a map of rare survival. Bone-rich sites drew attention because they could anchor an argument. Bone-poor sites often receded into uncertainty, even when tools, cut marks, ash, and animal remains suggested lives moving through them. Archaeology did not make a mistake in trusting bones. It built its confidence on the strongest evidence it had. But confidence can harden into habit, and habit can make the ground look quieter than it really was.
Long before archaeologists began treating sediment as human evidence, the ground was already preserving contact in another form. Not bodies, not artifacts in the usual sense. Residue. A person enters a cave and leaves more than footprints behind. Skin sheds. A hair catches and breaks. Blood sinks into dust. Waste seeps into the floor. Tissue decays. Fluids spread, ash settles, and the sediment takes all of it in, without caring what will matter thousands of years later. Most of that material disappears quickly. Some of it does not. What remains is damaged almost beyond recognition. Ancient DNA and sediment is not preserved as some hidden thread lying intact beneath the surface. It survives in broken fragments, chemically altered, and scattered through grains of dirt and mineral particles. Under the right conditions, that is enough. Cold helps. Stability helps. So does a protected environment where layers build slowly and are not constantly churned by water, roots, sunlight, or microbial life. That is why caves matter so much. Their floors can act less like exposed ground and more like gradual accumulation, storing traces from different moments of occupation in layers that look ordinary until they are sampled. DNA does not simply drift loose in the soil. Tiny fragments can bind to sediment particles, especially certain minerals, and that binding helps shield them from total destruction. Recovery depends on that kind of small physical shelter. What appears to be plain cave dirt may still hold molecular remnants in place long after bone has dissolved, scattered, or never survived there at all. None of this happens reliably. Preservation is uneven, often maddeningly so. One patch of ground may keep recoverable material, while another, only a short distance away, holds nothing. Heat, moisture, acidity, disturbance, and time itself all work against survival. Even so, the basic fact remains unsettling. A body can vanish from a site almost completely, and the ground may still retain the trace of having once been touched by it.
A cave layer that yields no human bone used to remain frustratingly unresolved. It might contain tools, a dark smear of ash, butchered animal remains, signs that something happened there more than once, and still leave the central question hanging: who had actually been present? Sediment DNA lowers that threshold. A layer no longer has to surrender a tooth or a fragment of skull before human presence enters the picture. In places where the old records stalled, the ground can begin to separate occupation from silence. That shift becomes most powerful in caves and other stratified sites, where use accumulated unevenly over long stretches of time. One level may preserve traces of carnivores dragging prey into shelter. Another holds firelight, stone flakes, broken bone, the residue of brief human activity. Above or below it, a faint genetic signal can tie the sediment itself to a human group, even when no body survived in that part of the sequence. The site stops looking like a single episode and begins to resolve into pulses: entry, absence, return, interruption, reuse, stacked one over another in a space that once seemed archaeologically thin. A shelter starts to look revisited rather than singular. A sequence that once seemed archaeologically sparse begins to separate into distinct occupations, long gaps, and returns. One hominin group appears in part of the record, another in a different layer. Animals and humans move through the same environment in patterns more complex than a few surviving skeletons ever suggested. What comes into view is not a gallery of individuals, but something archaeology has often struggled to recover from bones alone: a repeated rhythm of a place. The effect is subtle at first, then disorienting. Sites once treated as marginal begin to hold longer histories, and some of the old emptiness between the famous finds starts to look less like absence than like detail that had not yet become visible.
Denisova Cave had already changed the story of human origins before sediment DNA entered the scene. A finger bone, a few teeth, a handful of fragments, barely enough on their own to suggest the existence of an entire population. Yet from that sparse material came the recognition of the Denisovans, a group no one had known to look for. The discovery was extraordinary, but it exposed a familiar weakness in the archaeological record. Enormous conclusions were resting on a tiny number of surviving remains. The cave held more than those remains seemed to allow. Its deposits had built up across vast stretches of time, layer over layer, preserving evidence of repeated use. Tools, animal bones, traces of occupation were already there, but the biological presence of the people moving through those sediments remained frustratingly selective, visible only where chance had allowed a fossil to endure. Sediment DNA changed that threshold.
Genetic fragments recovered directly from the cave floor began identifying hominin presence in levels that had yielded no diagnostic human bone at all. The site changed shape. Neanderthal DNA appeared in some layers, Denisovan DNA in others, and the cave stopped reading like a place known through a few spectacular survivals. It began to resolve into a longer sequence of occupations, absences, and returns. Denisova was no longer simply a cave that had preserved rare bodies. It became a place where different human groups could be traced through time, even when their skeletons had vanished from view. That did not make the cave simple. Sediment DNA did not produce a neat tableau of coexistence, nor did it settle every question about who overlapped with whom and when. But it proved something the rest of the field could not easily ignore. A cave floor could preserve human history beyond the fossil record. And once Denisova made that plain, the silence of other sites became much harder to trust.
Denisova did more than illuminate one cave. It changed the standing of other sites that had long remained secondary. For generations, the story of human origins gathered around places that preserved bodies well enough to anchor an argument. Teeth, jaw fragments, bits of skull, these gave a site weight. Elsewhere, the evidence could be substantial and still feel second tier. Hearths, stone tools, butchered animal remains, long occupational sequences, yet no human skeleton secure enough to carry the same weight. The hierarchy looked natural only because bone survival had been mistaken for evidentiary importance. Sediment DNA begins to unsettle that order. In Siberian cave contexts associated with Neanderthal occupation, genetic traces recovered directly from deposits have helped tie particular layers to hominin presence where human bone was sparse, fragmentary, or absent altogether. Not every difficult site becomes clear. Many do not. Some remain stubbornly ambiguous, but the threshold changes. A place once read mainly through its artifacts can begin to acquire inhabitants again, and with that, older regional pictures start to shift. That is the larger shift Denisova forces into view. Archaeology has never worked from a neutral archive. It inherited a record filtered by preservation chemistry, burial conditions, scavengers, erosion, excavation history, and the simple luck of what remained. Sediment DNA does not erase those filters, but it cuts through some of them just enough to make the old distribution of evidence look less stable. A past that once seemed concentrated in a few exceptional caves begins to spread outward into places that had been harder to name. Some of those blank spaces will hold. Others may turn out to be products of survival bias more than genuine absence. That possibility changes the scale of the problem. The question is no longer only which famous sites preserve human origins best, but how many quieter places were pushed to the margins because the people who passed through them left no bones behind.
Sediment DNA can seem to require almost nothing. No skeleton, no tooth, no obvious human trace at all. That is also what makes it dangerous in the wrong hands. A genetic signal in a layer can be real and still be easier to misread than a fossil. Sediments are not static containers. Water moves through them. Burrowing animals disturb them. Later occupations cut into earlier deposits. DNA can migrate downward or sideways, linger unevenly, or survive in one patch of a site and vanish a few feet away. The archive is real, but it is not tidy. The most obvious danger is contamination. Modern human DNA is everywhere, on excavators, in laboratories, in the air of the present. Ancient human work has always had to contend with how easily the living intrude on the dead. Sediment samples demand even more caution because the evidence is already fragmentary. The signal is faint, damaged, and vulnerable to distortion before anyone begins interpreting it. A result can be technically authentic and still much less precise than the headlines built on top of it. Even when the DNA is ancient, presence is not the same thing as occupation. A trace may record a brief visit, decayed waste, a body passing through, or material introduced by later disturbance. It does not automatically reveal how many individuals were present, how long they stayed, what they were doing, or whether two groups actually met simply because their traces occur in nearby layers. Sediment DNA widens the field of vision, but it does not eliminate the need for stratigraphy, dating, artifacts, faunal evidence, and plain restraint. That restraint matters as much as the method itself. It is strongest not when it promises to replace the old archaeology, but when it forces archaeology to become harder to satisfy, more careful about absence, and less willing to confuse a quiet record with an empty world.
For a long time, archaeology treated survival as proof and disappearance as silence. If no bones remained, the past could look thinner than it really was, reduced to the rare places where bodies endured long enough to be found. Sediment DNA does not solve that problem so much as expose it. The ground can preserve contact after the body is gone, and with that comes a more unsettled picture of human origins, less clean, less evenly distributed, and less dependent on a handful of extraordinary fossils. Some of those blank spaces in prehistory will stay blank. Preservation still rules more than archaeologists would like. But some may turn out to be places where people were present all along, leaving traces too small for earlier methods to hear. The old record is not being overturned so much as made less certain, which may be the more important change. It leaves a harder question behind. How much of the human story has been mistaken for absence simply because we learned to trust silence too quickly? Armchair archaeology stays with the parts of the past that come back into view when old certainties begin to loosen. And if you want to continue with that same question in a closer human story, watch What We Got Wrong About Neanderthals and What We're Learning Now, where another past becomes harder to measure by what happened to survive. Until next time, keep questioning, stay curious, and remember, the record is never the same thing as the world it leaves behind.